Welcome to Justin Tutorials. Today we are going to talk about DNA replication in eukaryotes. In our last video, we talked about DNA replication in prokaryotes. So today we are going to talk about DNA replication in eukaryotes. If you can remember very well, in our last video, we made mention of some steps that are involved during DNA replication. We made mention of the initiation, we made mention of elongation, and we made mention of termination. So, same implies to the eukaryote. The DNA replication in eukaryote takes place in accordance with these three steps. The only difference is the same as prokaryote. The only difference are the DNA polymerase enzymes that are involved. Let's list the DNA polymerase enzymes that are involved. We have the DNA polymerase alpha, we have the DNA polymerase beta, DNA polymerase beta, we have DNA polymerase gamma, DNA polymerase gamma. We have DNA polymerase delta, DNA polymerase delta, and we have DNA polymerase epsilon. These are the DNA polymerase enzymes that are involved. The number one is the DNA polymerase alpha. This DNA polymerase alpha, it initiates RNA primer in both leading and lagging strand. DNA polymerase beta, DNA repair mechanism, it's involved in DNA repair mechanism. DNA polymerase gamma, mitochondria, the DNA replication. DNA polymerase delta, leading strand synthesis, and epsilon, lagging strand synthesis. We are going to name some of the proteins that are involved, the initiator proteins that are involved, as we go over to talk about the replication proper, where we come in contact with some of the proteins. So let's name them so before we can proceed. We have, number one of it is, these are the pre-replicative complex. The pre-replicative complex. We have the cell division cycle 6 protein. Second one is the cell division cycle 10 protein. The third one is the mini chromosome maintenance protein. The fourth one is the dependent transport 1 proteins. These are the pre-replicative complex that we are going to come across during the course of the DNA replication in eukaryotes. And also, let's acknowledge the sites where the replication is initiated. Let's clean this. You will appreciate this when we are done. The initiation process in eukaryote begins at multiple origin of replication. Multiple origin of replication. This multiple origin of replication is distributed along each chromosome called the replicator's multiple origin. Replicator's multiple origin. So, the initiation begins at the multiple origin of replication, distributed along each chromosome called replicator's multiple origin. There's a complex called origin recognition complex. Origin recognition complex. This complex binds to that site. This multiple origin of replication, that is, it serves as a site to which origin recognition complex binds. I said earlier that the multiple origin of replication is distributed along each chromosome called replicator's multiple origin. This replicator's multiple origin forms or creates the replication fork. So, the origin recognition complex binds to the multiple origin of replication. Now, the binding is not sufficient enough for replication to occur, but it is necessary. The binding of origin recognition complex is necessary for what? For the establishment of this pre-replicative complex. The cell division cycle 6 protein, the cell division cycle 10 protein, the mini chromosome maintenance protein, the dependent transport 1 proteins. You see, now this complex, it binds to the multiple origin of replication, which is distributed along each chromosome called the replicator's multiple origin. Now that binding process was necessary for the establishment of this pre-replicative complex. Now let's look at what will happen if this pre-replicative complex are established.